Have you ever wondered, truly wondered, just how many frogs are hopping, swimming, and croaking their way across our planet right now? The number is almost unimaginable. A testament to life's incredible tenacity. It's in the billions. A swirling, vibrant, global population of these ancient amphibians. From the tiniest thumbnail-sized peepers in your local pond to the giant, formidable Goliath frogs of Cameroon. But what if I told you that this vast, global chorus of frogs has been systematically and terrifyingly silenced over the last 50 years, their populations vanishing into a ghostly quiet, and that the key to unlocking this biological mystery, the patient zero of this ecological apocalypse, can be traced back to a single, unassuming, and frankly, quite goofy-looking frog from Africa. A frog that, in a bizarre twist of fate, became one of science's most celebrated heroes before revealing itself as the unwitting architect of one of the most devastating pathogens ever discovered. This is not a story of malicious intent, but a chilling tale of unintended consequences, a chain reaction that began with a quest for human knowledge and ended with a fungal plague pushing hundreds of species to the brink of oblivion. Our story starts not in a decaying rainforest, but in a pristine 1920s laboratory in South Africa, under the careful watch of a brilliant and forward-thinking scientist named Lancelot Hogben, a man deeply passionate about hormones and developmental biology, who was on a relentless hunt for the perfect model organism, a living creature. Simple and predictable, enough to help him unravel the complex chemical messengers that control life itself. Hogben wasn't a frog fanatic by trade. He was a physiological detective, and his case led him to a peculiar suspect, the African clawed frog, a creature so oddly adapted it seems almost alien, a flattened, slippery, tongueless aquatic frog that doesn't so much eat as it does vacuum its food, with unblinking, lidless eyes perched high on its head that give it a perpetually blank, thoughtless stare, a creature that can breathe through its skin and survive buried in dried mud for years, waiting for the rains to return. It was one particular, almost magical trick of this frog that captured Hogben's scientific imagination, its ability to change its skin color. Place it on a dark background, and over a few hours, its olive-green skin would darken to a mottled brown. Put it on a light surface, and it would slowly pale to a creamy white. Hogben had a powerful hunch that this color-changing superpower was governed by the pituitary gland, the master controller of the hormonal system. To test his theory, he performed a delicate surgery, removing the tiny gland from several frogs. The result was instant and dramatic. The frogs turned a ghostly, permanent white, no longer responding to their surroundings. He had proven his theory. The pituitary secreted a melanocyte-stimulating hormone that controlled the skin's pigment cells. But in doing so, he had accidentally created something far more valuable, a hormonal blank slate. These pale, pituitary-less frogs were now perfect, clean test tubes of flesh. They no longer produced their own internal hormonal chaos, which meant Hogben could now inject them with precise, known hormones from any source and observe the pure, unadulterated effects. It was a monumental breakthrough in endocrinology, and it was this precise setup that led to a discovery that would ripple across the globe, changing medicine forever. Some of Hogben's students, curious and experimental, decided to inject a small amount of ox pituitary gland extract into the dorsal lymph sac of these white, hormonally neutral frogs. They expected perhaps a minor physiological shift, but what they got was a biological spectacle. Within just a few hours, the female frogs, even without a male anywhere in sight, began ejecting hundreds upon hundreds of tiny, unfertilized eggs into the water, a process called ovulation that should only ever be triggered by the complex courtship rituals of mating. This was weird, it was gross, and it was utterly fascinating. But why was it such a world-altering big deal? Because of another substance, a common, everyday, human fluid that contains a very similar hormone to the one in the ox pituitary. Pregnant women's urine. You see, when a woman becomes pregnant, her body almost immediately begins producing a unique hormone called human chorionic gonadotropin, or HCG, which floods her system and is excreted in her urine. This hormone was already known to science, and researchers had already developed other animal-based pregnancy tests, like the infamous rabbit test or mouse test, where a woman's urine would be injected into a young female rabbit or mouse. If the woman was pregnant, the HCG would cause the animal's ovaries to become enlarged, a result that could only be confirmed by killing and dissecting the poor creature days later, a process that was slow, expensive, morally questionable, and sometimes inconclusive. But Hogben's frog, the African clawed frog, changed everything. The Xenopus pregnancy test, as it became known, was a miracle of modern diagnostics. It required just one single injection of a woman's urine into the frog's back. There was no waiting for days. If the woman was pregnant, the HCG in her urine would mimic the ox pituitary hormone, and the frog would reliably lay its eggs within 8 to 12 hours. The results were clear, objective, and visible to the naked eye, no dissection required. The frog didn't have to die. It could be kept in a tank and used again and again, making the test more humane, vastly cheaper, and over 98% accurate. When this test was formally published in 1938, it revolutionized reproductive health care, empowering women with faster, more reliable information about their own bodies. The demand for African clawed frogs exploded overnight. They became a global laboratory commodity. South African suppliers scoured ponds and rivers, collecting them by the tens of thousands, shipping them in specially designed containers to hospitals and research labs from London to New York, and eventually to every corner of the globe, South America, the Middle East, Asia, and Australia. By the 1950s, the African clawed frog was one of the most ubiquitous and important laboratory animals on Earth, a true hero of science, a slimy, unblinking savior for countless women. But this golden age of the frog test was not to last. By the 1960s, science had advanced once again, developing the elegant pea-on-a-stick immunochemical test we know today, which uses antibodies to detect HCG without the need for a living animal. The frogs were suddenly obsolete. Laboratories around the world now had thousands of these resilient amphibians, with no further use for them. Many were euthanized, but many others were simply released into local waterways by well-meaning pet owners or lab technicians, or they escaped from breeding facilities. The African clawed frog, a hardy survivor, thrived in its new homes across the Americas and Europe, becoming a successful invasive species. And for a while, it seemed like the story was over, a happy ending where technology had progressed, and the frogs had found a new lease on life in foreign lands. But this was not the end. This was merely the calm before the storm. Lurking on the skin of these globe-trotting frogs, hidden from view, undetectable by the science of the time, was a sinister, silent stowaway, a biological time bomb that was about to unleash hell on the world's amphibian populations. The first terrifying signs of this emerging catastrophe appeared away from Africa, in the pristine, upland rainforest near Brisbane, Australia, in the late 1970s. Researchers conducting routine amphibian surveys stumbled upon a scene from a nightmare. Streams that should have been teeming with life were instead littered with hundreds of dead and dying frogs. Among the casualties were some of Australia's most unique species, like the extraordinary gastric brooding frog, the only frog known to incubate its young in its stomach. Finding a single dead frog in the wild is rare. They decompose quickly or are scavenged. Finding hundreds was a cataclysmic red flag. The scientists were horrified and baffled. They monitored the populations closely, but it was too late. By 1981, the southern gastric brooding frog had vanished entirely, gone from the earth forever. Then, a terrifying pattern emerged. A wave of death began rolling northward through Queensland's rainforests at an alarming pace of about 100 kilometers per year. In the mid-1980s, it hit the jungles of Ungella, wiping out the northern gastric brooding frog and devastating the Ungella day frog. By the late 1980s, the far northern rainforests were struck, with at least 7 species experiencing catastrophic declines. 4 vanished completely. The crashes were shockingly swift, often wiping out entire populations within a single season. This was not a slow, gradual decline from habitat loss. This was the signature of a highly virulent, fast-moving disease. A 1994 study provided the first crucial clue. Scientists collected healthy frogs and placed them in 2 separate tanks, one with clean rainwater and one with water from the infected streams. The frogs in the rainwater thrived. The frogs in the stream water died within days. The killer was in the water. But what was it? A virus? A bacterium? The answer remained elusive. Then, in the 1990s, an identical nightmare began playing out an entire ocean away, in the montane rainforests of Panama and Costa Rica. Herpetologists there documented the same horrifying scene. Streams filled with dead frogs of multiple species. A rapid multi-species die-off with no obvious cause. Scientists from Australia and Central America joined forces, sending tissue samples from their dead frogs to pathologists. Under the microscope, after ruling out countless other possibilities, they finally found the culprit. It wasn't a virus or a bacterium. It was a fungus. Embedded deep within the skin of the dead frogs were the reproductive structures of a previously unknown aquatic chytrid fungus. In 1998, the disease was named chytridiomycosis, and in 1999, the fungus itself was identified as Batrachochytrium dendrobatitis, BD for short. BD is a nightmare pathogen. It is an aquatic fungus which is rare in itself, and it is obligately parasitic, meaning it can only live and complete its life cycle on the skin of living amphibians. This is its only home. It produces microscopic, free-swimming spores called zoospores, each equipped with a single whipping tail, allowing it to swim through water like a tiny torpedo, hunting for amphibian skin. Once it finds a host, it burrows in, grows and forms a sporangium, a sac that matures and produces hundreds of new zoospores internally. When the sac ruptures, it releases a new army of spores onto the frog's skin and into the water, ready to infect new victims. For the frog, this infection is a torturous death sentence. A frog's skin is not just a covering. It is a vital organ for respiration, water absorption, and electrolyte balance. As the BD infection spreads, it causes the skin to thicken dramatically, sometimes by 40 times its normal width. It becomes leathery, peels away, and loses its function. The frog cannot breathe properly. It cannot maintain its critical water and salt levels. Its heart rate slows and it essentially suffocates or experiences cardiac arrest, often sitting listlessly in the water before dying. The fungus thrives in the cool, moist conditions of upland streams, which is why the first die-offs were seen in high-altitude rainforests, the very places that served as pristine refuges for so many unique frog species. The spread was relentless. As scientists raced to understand it, BD swept across the Americas, Europe, and Asia, driving massive declines in over 700 amphibian species and causing the confirmed extinction of at least 90. This is not like the fictional fungus in The Last of Us. This is worse. Imagine a single disease that could infect nearly every species of mammal on Earth, every human, dog, cat, cow, and monkey, with a near 100% fatality rate. For the world's amphibians, the chytrid fungus is that apocalypse. The burning question remained, where did this monster come from? The puzzle pieces were all there, but it took until 2004 for scientists to definitively put them together. A team began a massive forensic investigation, examining preserved museum specimens of amphibians collected from all over the world from the early 1900s onward. By analyzing snippets of skin, they could test for the presence of BD DNA. The results were shockingly clear. The earliest positive cases, dating back to the 1930s, all came from one place and one species, Africa, on the African clawed frog. The timeline was a perfect match. The global spread of BD directly followed the global trade of the African clawed frog for pregnancy testing. The reason it had gone unnoticed for decades was that the African clawed frog, having evolved alongside the fungus for millennia, was largely immune. Its skin produces powerful antimicrobial peptides that keep the fungus in check, allowing it to live as an asymptomatic carrier, a walking reservoir for a pathogen that was utterly lethal to naive amphibian populations with no evolutionary history with it. When these lab frogs were released or escaped into new environments, they didn't just become an invasive species. They became patient zero in countless new outbreaks, unleashing the plague upon the world. The road to hell, as the saying goes, is indeed paved with good intentions. Lancelot Hogben, a man who believed science should serve all of humanity, who fought for better public health and women's reproductive rights, had inadvertently set in motion an ecological disaster of staggering proportions. This was not a story of corporate greed or moral failure. It was a colossal, tragic accident, a reminder of the profound and often unpredictable interconnectedness of our world. Now, knowing the enemy, scientists and conservationists are waging a global war to save what remains of our amphibian diversity. The challenge is immense, as bead is now permanently established in many ecosystems. But human ingenuity, the very force that accidentally caused this crisis, is now being marshaled to solve it. One front involves direct treatment, like giving captive frogs medicated antifungal baths. These can clear the infection temporarily, but they are impractical for wild populations on a large scale. Another, wonderfully creative, approach involves using the pathogen's own weakness, its sensitivity to heat. In a brilliant 2024 study, scientists built simple, cheap frog saunas, small, miniature greenhouses made of bricks and black roofing tin that heat up in the sun. They found that infected frogs, seemingly understanding the relief, would voluntarily enter these saunas, raising their body temperature, enough to significantly reduce their fungal load and even clear the infection entirely, giving them a fighting chance to survive and build up a natural immunity. Perhaps the most promising long-term strategy lies in the frogs themselves. In populations that have been hammered by BD, a few resilient individuals often survive. Scientists are now studying these survivors, identifying the genes, immune responses, and beneficial skin bacteria that confer resistance. The goal is to use this knowledge for targeted conservation breeding programs, releasing resistant frogs back into the wild to re-establish populations that can live alongside the fungus. Solving the Chytrid crisis is a monumental task that will require a global effort, combining field biology, genetics, ecology, chemistry, and data science. It is a stark reminder that while human progress can have devastating, unintended consequences, it is also through science, dedication, and a profound respect for the natural world that we can hope to mend the fractures we have created. The story of the African clawed frog is a cautionary tale, but it is also a story of resilience and redemption, a testament to the fact that saving our planet's incredible biodiversity doesn't just require compassion. It demands unwavering scientific curiosity and the relentless determination to solve the problems we ourselves have created. If you enjoy this video, please like, share, and subscribe our channel Animals Biography. And tell us which part did you like. Thanks for watching.